All right, final lecture in this little, mini lecture series is on Mendelian genetics, is advanced genetics concepts. We have talked about monohybrid crosses, dihybrid crosses, sex linkage, incomplete dominance, codominance, multiple allelic inheritance, gene linkage, and sex linkage. So we've gone through a whole lot of information. Now we're getting into the advanced genetics concepts that go beyond Mendel. One of them is pleiotropy. Pleiotropy is where a mutation in a single gene can have multiple effects throughout the cell. So one gene, multiple effects. One example would be microtubule formation. Microtubules are used for many different things. They're used for cell division. They're used for moving material around the cell. And they're used for a little bit of cell shape. So one gene can have an impact on multiple different aspects of the cell. With microtubules, cell cycle, and cell movement. Different cells with pleiotropy can express the same gene in different ways. In this particular example, the same gene that controls the removal of the webs between fingers during development, also controls the formation of dentition in the mouth. One gene, a change in one gene, caused both of these effects. That's pleiotropy. The same gene might be expressed at different times during development in different ways. The same gene that's responsible for, say, determining left side of your body from the right side of your body might be also responsible for limb formation. Now, in snakes, obviously, early snakes had limbs, but that gene's function at one point in its development is turned off. So a gene can do different things at different times during development. That's pleiotropy. One gene, multiple effects, whether it's at different times in development or simultaneously in one cell or expressed in different cell types in different ways. We're also going to talk about epistasis. Epistasis is where the allele of one gene masks the phenotype of a different gene. For example, hair color, right? In this one, you could have red hair color or blonde hair color. You might have one gene that's responsible for either of those. Now, hair color is polygenic, so this is simplifying it a lot. The deal is, though, in order to see either of those, you need to have the gene to produce hair. You might have the gene for red hair color, but if you don't have the make hair gene, you will never see it. That phenotype is hidden. So you have one gene that can, on one gene on one chromosome, will hide the effect of another gene on a different chromosome because it may be required part of a pathway. It might be the first gene product in the pathway that you need that something else can build on. And if you don't build that initial gene, you're not going to be able to change it later with other enzymes. So epistasis is where one gene masks the phenotype of another gene. This can occur during embryonic development. One gene or one chromosome might get turned off entirely for that individual's life. It doesn't impact the offspring because that gene will basically come back alive during meiosis and mitosis. But for the life of the cell outside of reproducing, one or another gene, one or the other homologous genes are turned off. So two big examples of this are X chromosome inactivation and genome imprinting. With X chromosome inactivation, women have, in humans, two X chromosomes. That means that they would normally produce twice as much protein as a male would. They've got two chromosomes that are fully working compared to a male's that's only got one working. Well, the solution that nature's come up with is not double how much the male's X chromosome has to produce, but turn one of the X chromosomes entirely off. So during the cell's life, one of the two X chromosomes, entirely random which one, gets turned off. And when it's turned off, it's able to, it sort of forms this little ball. It just curls up into a ball and hides in the corner of the nucleus. And it's called a Barr body. Nothing happens with it. It's turned off. When the cell starts to go into mitosis and meiosis, that X chromosome unwinds and is able to be replicated, and then in the daughter cells, one of the two X chromosomes is entirely turned off. Totally random. With genomic imprinting, instead of turning off an entire chromosome, sometimes either one or the other of the genes, one of the or the other of the homologous pairs is turned off. You might only express like the gene from your mom or the gene from your dad, depending on what it is. So the offspring are expressing only one of the two alleles, regardless of whether they are homozygous or heterozygous. And here's an example for growth factor. If you delete the father's IGF, so the father's IGF is entirely turned off, and the mother's is on, you get a teeny tiny mouse. And that teeny tiny mouse comes with a whole bunch of siblings. So lots of siblings, very small mouse. If you deleted the mother's IGF, you end up with gigantic mice that have very few siblings. So imprinting will turn off usually it's one or more genes, whereas X chromosome inactivation turns off an entire chromosome. So that's epistasis. The allele of one gene masks the effect of another, regardless of what that other one is. Another concept I want to cover is polygenic inheritance or polygenetic inheritance. Polygenic inheritance is almost the exact opposite of pleiotropy. It's many genes having one effect. Typically, these are going to be genes that have what's called a gradation. They have a wide variety of intermediate forms. So many genes contribute to the outcome of a phenotypic trait. You can have two types of traits in science. You can have discrete traits, tall and short, yellow and green, axial and terminal. Or you can have quantitative traits. Quantitative traits show variation among a range. So you've got height, eye color, hair color, where it's just not tall, short, but it might be, I don't know, 5'1", 5'2", 5'3", 5'4", 5'5", 5'6", and all the numbers in between. There's a lot of variation. Mendel described discrete traits. Polygenetic inheritance describes quantitative traits. Quantitative traits are usually polygenetic, and they are heavily influenced by the environment, by what you eat, by what you drink, by how much sun you get, and all of the other such. So polygenetic traits are multiple genes contributing to the outcome of a single trait. They tend to have large gradation, a large range, and they are highly impacted by environmental conditions. And then finally, we have extranuclear inheritance. There is DNA outside of the nucleus, and it lives in the mitochondria and in the chloroplast. Because remember that whole endosymbiosis thing we talked about a while ago, where perhaps a small organism got taken in by a larger organism, and then they live together in harmony? These genes are these mitochondria and chloroplasts are usually maternally inherited, and that's really because there's not many of them that will fit inside of a sperm. Sperm are really small. The egg, gigantic and filled with the machinery of life. So because each of those possessed its own unique DNA, it's going to be inherited along the matrilineal line. And that's super neat because we can actually use mitochondrial DNA to trace back our ancestry through our mother's line, and that lets us track human migrations from the earliest humans evolving on the planet, moving out of Africa in multiple waves. So extranuclear inheritance, any DNA that's inherited from outside the nucleus, specifically in the mitochondria and the chloroplast. And it will follow the mother's line more than the father's line. So yeah, genetics isn't nearly as easy as Mendel made it out to be. There's a lot of these advanced concepts that don't follow Mendel's rules. And we're still learning more about it day by day. We talked in this particular lecture about pleiotropy, one gene, multiple effects. Polygenetic inheritance, many genes, one effect. Epistasis, one gene masking the effect of another. And extranuclear inheritance. Anytime you have DNA from outside of the nucleus that's inherited, usually in the form of chloroplast or mitochondria. These content review questions are here to focus your studies. In the next set of mini lectures, we're moving beyond predicting um, what will happen in the offspring to talking about the molecular mechanisms associated with the central dogma of molecular biology: transcription, translation, and replication. How do we copy our DNA? How do we read the DNA to make RNA? And how does that RNA then get read by a ribosome to produce a polypeptide? Once we go through that very, very important set of lectures that forms the basis of modern biology, you'll have a strong grounding for your future biology courses.